Good morning. Thank you very much for joining us today to one more webinar of the NewTechAqua webinar series. Today we are here for the webinar about reproduction of broodstock management of Meagre, Argyrosomus regius. Just before we start, let me remember you about the NewTechAqua project. So NewTechAqua project is a H2020 project funded by the European Union. It is an innovation action, meaning that the outputs, the knowledge generated, should reach the industry. And it's a project that was approved under an ambitious call that was launched in 2018 to 2019, called Sustainable European Aquaculture 4.0 Nutrition and Breeding. Under this call, 4 projects were approved. The 4 of them are arriving, they had finished or arriving to its end. They are the e-efficiency, intelligent fish feeding through integration of enabling technologies and circular principle, aqua impact, genomic and nutritional innovations for genetically superior farmer fish to improve efficiency in European aquaculture, NewTechAqua, new technologies, tools and strategies for a sustainable, resilient and innovation in European aquaculture, and future EU aqua, future growth in sustainable, resilient and climate-friendly organic and conventional European aquaculture. So, about NewTechAqua, new technologies, tools and strategies for a sustainable, resilient and innovation European aquaculture. Just to let you know that it's a project that it has an overall budget that close to 7 million, 6 million coming from the European Union and the rest from some of the partners on the industry. It's an innovation action. It is a project of 4 years of duration that started in January 2020 and is finishing at the end of this year. It's a consortium that is coordinated by the University of Bologna and is counted on 25 partners from 9 countries: Belgium, Croatia, Cyprus, France, Greece, Italy, Norway, Spain, and United Kingdom. So the consortium it has partners that come from research and development, research institutes, universities like UNIVO, University of Venezia, IRTA, HCMR, University of Bari, NOFIMA, IFMR, CISAF, DFMR, University of Las Palmas de Gran Canaria, and CNRS from Italy. And also 12 partners coming from the industry, either from aquaculture companies, fish farming companies, 4 biotechnology companies from aquafeed producers, and 3 more partners that are working on exploitation and disseminations, like the Federation of European Aquaculture Producers, SIGEAN, which is the institution I belong to, and we are the one coordinating all training, knowledge transfer activities, such as these webinars, and Green Innovate from Belgium. So what is the objective of the strategy of NewTechAqua? Well, it is to expand and diversify aquaculture production of fin fish, mollusks, and microalgae by developing and validating technologically advanced, resilient, and sustainable application. This is a project that has focused the research in the main important fish species produced in Europe, such as salmon, trout, sea bass, and sea bream, but also has worked in emerging new species like amberjack, Meagre, sole, and mallet. And besides, also work on mollusks like oysters and mussels, and microalgae species. Those are the species that the project has focused its work, but it has done it working on different disciplines, like industry, artificial intelligence, precision farming, on production of new feeds, more sustainable circular aqua feeds, new farming technologies like aquaponics, development of new products, or the optimization of the production of emerging new species, also in genetics and breeding, in order to provide integrated solution for a more sustainable and more efficient aquaculture. So just we are at the end of the project, and an important part of our work is to do the maximum dissemination as we can about the outputs and the knowledge generated by the project. And for that, we have worked on different activities, such as the organization of advanced courses. We did a course on nutrition and feeding here in Zaragoza, by CGE and Zaragoza organized. And this week is taking place in Tricase in Italy, organized by our colleagues from CGE and Bari, another course about organic aquaculture. We have also organized one workshop about low environmental impact aquaculture in the month of June in Spain with IRTA in Tarragona. And next week, there is a workshop organized by our colleagues of Bari about precision farming in land-based aquaculture facilities, which is going to be organized in Bologna. We are developing e-learning modules. We are organizing webinars, as the one that we are organizing today. And finally, our last activity is going to be the school campaign. We want to go to the schools to disseminate to teenagers these advances that aquaculture is implemented thanks to the research, and in order to provide a more sustainable image of aquaculture. Well, today we are in one of the webinars that we are organizing during the month of October, which is our more intense month, is the webinar about reproduction and broodstock management of Meagre. If you had missed one of the previous webinars, just take note of this address, because we are recording all of them and they are available after the webinar has taken place in the YouTube channel that we have. Just to finish about the dissemination activities we have, just to let you know that 15th of November we are joining in Brussels in the final conference to share with those that will attend the main outputs of the project. So maybe Dinos, if you want to take the floor, please, you can introduce this webinar.
Okay, good morning from myself. I'm Constantinos, or Dinos Mylonas. I'm from the Hellenic Center for Marine Research, the Institute of Marine Biology, Biotechnology and Aquaculture. And I was the work package leader for the reproduction and closing the cycle of new emerging species. The work package consisted of 3 tasks, one for Meagre, Argyrosomus regius, one for the greater amberjack, Seriola dumerili, and one for the Senegalese sole, Solea senegalensis. Today we're presenting our work on Argyrosomus regius, on Meagre. And for Meagre, our task was to try to look into, first of all, environmental management in order to enhance the reproduction and our control of reproduction in captivity, and also use hormonal methods to enhance control and also advance puberty. The problem we have with Meagre in aquaculture is that although it undergoes gametogenesis without any problem, and males complete gametogenesis and produce viable sperm, in females, very often we do not observe the occurrence of final maturation, of oocyte maturation, and therefore no ovulation, no spawning. Therefore, the majority of hatcheries in the Mediterranean require hormones to induce maturation and to induce spawning. And we wanted to try to learn as much as possible about the reproductive biology and behavior of this fish in order to overcome this problem, mainly from the point of welfare for the fish, and to try to make the process as natural as possible, but also because when you want to produce fish in an organic procedure, you're not allowed to use any hormones, so there's no possibility to produce organic Meagre right now. The presentations we chose for you today are shown in the next slide. We will start with a description of the process of gametogenesis of wild Meagre and rare and different conditions. Then we will continue. This is a presentation from the University of Bari. It will be presented by Dr. Rosa Zupa. Then we'll have a presentation from a PhD candidate we have here at HCMR, Stefano Lancerotto. He's going to talk about our efforts to use salinity and temperature manipulations to enhance maturation and spawning, allow spawning without hormones in Meagre. I will then take the floor and talk to you about the breeding behavior of these fish and their relation with sound production and the spawning kinetics and dynamics in terms of parentage contribution. And we'll finish with Neil Duncan's presentation from IRTA in Spain, where in collaboration with Rara Avis and Ignacio Jimenez and his colleagues, they produce recombinant gonadotropins with the objective of enhancing gametogenesis, but also inducing it in pre-pubertal fish to advance puberty. So this is what I had as an introduction. We'll have a questions and debate after the presentations, and I will give the floor to Bernardo back to explain you how this will be done.
Yeah, thank you, Dinos. So for the last part of the webinar, for the questions and for the debate, the Zoom has a tool that is called questions and answer. But it also has another tool that is the chat. Most people is familiar with the chat, but today you are invited to write your questions using this tool, because there we can have conversations related with an inertial question. In the chat, you can use it maybe to maintain conversations among you or others, but not for the questions. For the questions, please use this one. So then I think that now we can start with the first presentation of Rosa Zupa. I'm going to stop sharing the screen and asking Rosa Zupa to share your screen, please.
Okay. Thank you, Bernardo. Thank you, Dino. So I'm going now to share my presentation. Okay.
Okay, now we see
Can you see?
Yes, but okay. We see it well. You can go ahead.
Okay. So good morning, everyone. I'm Rosa Zupa from the University of Bari in the south of Italy. And okay, so my main research interests are focused on fish reproduction. These are my contact details, so if you have any question and information, you can contact me. So today I'm going to talk to you about the Meagre gametogenesis in the wild and in different rearing conditions. In particular, I will describe you how is made an ovaries and the testes of Meagre, and the different stages of gametogenesis of Meagre, that is the oogenesis and the spermatogenesis. And then I will provide you with basic elements on Meagre reproduction in the wild. And then we'll see the effect of rearing in captivity on Meagre reproduction. So first of all, why we need to compare the reproduction of Meagre in the wild and farming condition? Because the knowledge of the gametogenesis process is important, is necessary for the reproduction control in aquaculture. This is for all the fish species. So let's see now how is made the ovary of a Meagre. The ovary of Meagre, as the ovary of other fish species, is made of is a pale elongated organ, which is located in the body cavity. This organ is thin when fish is young, when fish is juvenile. And then in adult fish, it increases in size, in length, and also changed color during the reproductive cycle as maturation proceeds. You can see here, this is a micrograph of an ovary of a juvenile meagre. The ovary is surrounded by a musculary ovary wall and consists of ovigerous lamellae, which are this, which contains the oocyte at different stages of development. In the inner part of the ovary, there is a virtual cavity, which in adult fish became larger, and is called lumen. And in the lumen, the oocytes are then ovulated. The meagre is a multiple spawner fish. This means that during the reproductive cycle, the meagre spawn release in the water several batches, group of eggs, a certain number of batches of eggs. And you can see from this picture, 2 different batches, groups of eggs at different stages of development. These are less mature and these are more advanced oocytes. In general, as a general definition, the oogenesis is the process in which diploid germ cells called oogonia proliferate and differentiate to form eggs. This is a picture of the oogonia of a meagre. These cells are the smaller cells in the oogenesis and are present in the ovary throughout the fish life. In the meagre, these cells range between 9 and 17 micrometer. The oogenesis is divided in 3 phases. The first phase is called primary oocyte growth, and this phase is present in both juvenile and adult fish. In this phase, the oocytes are at the perinucleolar stage. In this stage, there is a very high level of protein synthesis. It's very active. In the second phase is defined the secondary oocyte growth. This is a phase which occurs only in adult fish because it requires the activation of the reproductive axis. This second phase is characterized by vitellogenin uptake and yolk accumulation. Vitellogenin is a protein which is synthesized by the liver and then is transported to the ovary throughout the bloodstream. It reaches the oocyte and start to be incorporated as yolk granules. You can see here these globules, which are the yolk granules. And these are 2 different stages that we can observe during the vitellogenesis process. Early vitellogenesis stage in which the oocytes start to be filled with yolk granules, and then this is the more advanced stage of vitellogenesis. We find the late vitellogenesis stage of the oocyte in which the yolk accumulation reach its maximum level. Of course, this phase is the most important during gametogenesis because yolk will be the nutrient for the embryo. And so you can understand that is very important that it occurs in the proper way. The third phase is called oocyte maturation. And this is characterized by the resumption of meiosis of the oocyte, of course, and then the nuclear migration and breakdown. This phase then is followed by the oocyte hydration, in which the oocyte increases in size because it's filled with water. Both these stages are very short. It's difficult to observe in the ovary of fish. In fact, we we were not so lucky to see these two stages in the ovary of a Meagre. These two pictures are from another fish species, just to show you how these two stages appear. After oocyte duration, the oocyte is then ovulated. This means that the oocyte is released in the lumen, and so the oocyte is surrounded by follicular layer. And so the follicular layer open, and then the oocyte is released, and then the follicular layer remains in the ovary. You can see here, this is called post-ovulatory follicles. And also this post-ovulatory follicles is a very short life. So the presence of the post-ovulatory follicles in the ovary indicates that the fish has spawned recently. So during the normal gametogenesis, some oocytes in vitellogenesis died. This process is called atresia, which consists of many events, including the old reabsorption. You can see here nitritic follicles in advanced follicles, in late vitellogenic follicles. This is a physiological process which occurred during gametogenesis, but the presence of most or all vitellogenic follicles in atresia may indicate the occurrence of reproductive dysfunction in the ovary of the fish. Meagre females reach sexual maturity at 4 years of age. So here you can see an ovary of a juvenile fish, 18 months of age, in which there are only oogonia and primary growth oocytes. In this picture, you can see an ovary from an adult fish, of course, with more advanced oocyte stages. Let's see now how is made testes of a Meagre. As for the ovaries, the testes are pair elongated organs located in the body cavity. And also in adult fish, the testes increase in size, length, and also change their color during the reproductive cycle as the maturing process proceeds. The testes of Meagre and generally of all fishes consist of seminiferous tubules. You can see the seminiferous tubules in this picture, the higher magnification. The seminiferous tubules are extended from the periphery of the testis to the inner part of the testis and are made of a wall which contain the germinal epithelium with the different germ cell stages. And in the center, each seminiferous tubules is made as a cavity, which is called lumen, in which the spermatozoa are then released. And then all the spermatozoa released in the lumina of the seminiferous tubules are conveyed in the main sperm duct. The spermatogenesis is the process in which diploid germ cell spermatogonia proliferate and differentiate to form spermatozoa. Male germ cell in fish develop inside cysts. A cyst is made by somatic cell called Sertoli cell, which surround synchronously developing germ cell clone deriving from a single spermatogonia. So you can see here a single spermatogonia surrounded by the germ cells, which start to proliferate and then enter the first meiotic division and become primary spermatocytes. And you can see a cyst of primary spermatocytes in a Meagre testis, which is this. Then, during the first meiotic division, primary spermatocyte became then secondary spermatocytes. You can see here a cyst of secondary spermatocytes. And then secondary spermatocytes enter the second meiotic division and became spermatids. You can see here. Okay, this is a cyst of spermatid. And then spermatid differentiate to form spermatozoa. this is a cyst containing spermatozoa, which then open, and so the spermatozoa are released in the lumen. And this is a lumen of a seminiferous tubule filled with spermatozoa. Males in Meagre reach sexual maturity at 3 years of age. This is a testive of juvenile fish with small seminiferous tubules containing the mainly spermatogonia or the less advanced stages of spermatogenesis. And this is a testis from an adult fish, adult Meagre, in which you can see all the stages of spermatogenesis and also spermatozoa in the lumen of the seminiferous tubules. So let's see now the basic elements of the reproduction of Meagre in the wild. We don't have so much information regarding the Meagre in the wild. There are only a few studies related to 2 different areas. This area, which is the Mediterranean Egyptian coast, and here the Gulf of Cadiz in the Eastern Atlantic. From these 2 studies, we know that Meagre gametogenesis starts in March, April, at the temperature of about 16 degrees. I have to say that the temperature is very, very important to trigger gonad maturation at the beginning of the reproductive cycle of a fish species. And then the active gametogenesis and the spawning occurs until July in the Mediterranean and until August in colder water of the Eastern Atlantic. As I told you before, the fish gonads undergo changes in weight and appearance during the reproductive cycle. And this change can be expressed as relative gonad mass, which is the gonadosomatic index, GSI, which is the ratio expressed in percentage between the gonad weight and the body weight of the fish. And is a good indicator of the reproductive maturation. From these 2 graphs, you can see the trend of the GSI in females and males, in wild females and males Meagre in the Mediterranean. And you can see that the GSI reached the peak in females in April, while in males reached the peak in March. And then these values start to decrease as fish start spawning. How many eggs does Meagre release in the wild? The number of eggs is expressed as fish fecundity, which is related to fish size. Larger fish release more eggs. We know that from the literature, Meagre female of a body weight of 1 kilo produce about 1 million eggs for each reproductive season. Let's see now what happened to these fish when they are reared in indoor concrete tanks. There are 2 studies that were carried out in 2 different experimental farms. The first study was carried out in Spain on Meagre caught from the wild and reared in land-based tanks. And this study reports that these fish didn't reproduce spontaneously. The other studies that was carried out in Crete on Meagre born in captivity, so called hatchery produce, and reared in land-based tanks, reports then part of females were able to reproduce spontaneously with an estimated fecundity which was quite lower than what is reported for wild population. Hormonal treatments were effective in stimulating oocyte maturation and spawning in this fish, and also in increasing fecundity of this fish. What about Meagre when it's confined in floating sea cages? We did a study in a fish farm in the south of Italy on Meagre born in captivity and reared in floating sea cages. And we saw that the temporal pattern of gonad maturation seemed to be quite delayed compared with wild population. This means that you can see from these two graphs, the GSI of females and males living in floating sea cage peak in June compared to what happens in the wild population, in which the peak occurred in March, April. However, these fish were able to finalize the reproductive maturation process. This is an ovary of a female, of course, reared in floating sea cage with advanced oocytes. And this is a testis of a Meagre reared in floating sea cages, which was mature. You can see here plenty of spermatozoa in the lumen of the seminiferous tubules. Does this fish born in captivity and reared in floating sea cages ovulate spontaneously? Yes, because we observed post-ovulatory follicles in the ovary together with other signs of ovulation. But we didn't look for eggs in the cages. So what can we conclude for all these data that we have? That Meagre caught from the wild and reared in land-based tanks seem to not reproduce spontaneously. Better adaptation to confinement seem to have Meagre born in captivity and reared in land-based tanks, since, as we have seen, these fish were able to finalize the gametogenesis process spontaneously. Promising results came from Meagre reared in floating sea cages in terms of their capacity, their ability to reproduce spontaneously. What can we suggest? What are our recommendations about these two rearing conditions? Regarding the land-based facility, maybe improve the rearing condition and reproduction control protocols could increase the egg production and fertility at the same level as wild population. Regarding the sea cages, we need to do further studies to confirm the capacity of this fish to spawn spontaneously. And also we need to collect eggs to evaluate their quality, their fertilization rate, their amount, so to evaluate fertility in this fish. So I finished. This is a list of reference. If you need, I can give you more information about this. And for any question, you can write your question in the Q&A tool. Thank you so much for your kind attention.
Okay, thank you so much. Well, thank you very much, Rosa. Then let's move to the next presentation, Stefano Lancerotto from the Lenix Center for Marine Research, please. And as Rosa said, if you had question doubts, you can already start writing them in the questions and answer tool. So, Stefano.
Thank you, Bernardo. Good morning, everybody.
Good morning.
And start sharing the presentation. So, as part of this webinar, this presentation is about the effects of salinity and temperature. And the study was
Stefano, you haven't shared the screen yet.
Oh,
no?
okay. Why not? Just a second, please. Okay. Can you see that now?
Yes. Yes.
Okay.
Go ahead.
Okay. So I was saying the study was conducted in the HCMR by the research group present in the Aqua Lab. And the target was mainly as a service for the aquaculture industry in a way that the effect of environmental parameters can ameliorate the egg production and therefore increase the production itself of Meagre. The first study that we conducted was related to the importance of temperature in gonadal maturation and egg production and quality. The importance of thermal cycling, why? Well, as Rosa said before, Meagre is a temperate species, so is influenced during the maturation by temperature as one of the main factors. And for this reason, we decided to maintain 2 broodstock and imposing on each one of them a different thermal regime. As you can see from the graph, one was constantly maintained at 19 degrees, more or less. This is why in the Mediterranean, many hatcheries can decide to supply the stocks of fish with borehole water, with comes it's safer in terms of biological supply of organism or microorganism. So the only disadvantage is that this water has a constant temperature, so it has to be cooled down or warmed up, so increasing the expenses. The second stocks was actually was imposed to it a temperature that was decreased during the winter, so giving, as Rosa said, maturation start when from a certain temperature, it starts increasing during March, April, and May. So we decided to decrease the temperature water during the winter and gradually increase it again during spring. After that, fish were evaluated, obviously, were assigned, were administered with GnRH for spawning induction, as in land-based tank, they cannot reach maturation themselves. And then the production of eggs was evaluated together with larval quality after hatching. Okay, so first, starting with the male, our results said that imposing a constant temperature, so denying a winter seasonality, even if slightly reduced in terms of amplitude of the winter season, negatively affected the capacity of males to complete gametogenesis and freely release males. While on the other side, males that underwent a winter season, they were found already releasing sperm. After the GnRH implantation, this permeation capability increased gradually for both groups. However, as you can see from the right part of the graph, the constant group did actually not ameliorate the situation of spermiation that much, meaning that obviously fertilization couldn't be achieved in the same terms. On the side of the female, the situation was a bit better. In both group, females could reach advanced vitellogenesis. However, you can see from both ovarian biopsies and histological section that the percentage of advanced vitellogenic oocytes in the two groups are quietly different. And also the diameter of the oocyte didn't reach what is recognized as the minimum values that is optimal for the administration of GnRH. This is translated in a decreased production in terms of relative fecundity, so the quantity of eggs per kilos of female. And this one can be seen in the graph on the left. And the same was also reflected in the survival capability of the embryos, not after hatching, but during the 24 hours after spawning. So the development of the embryo was negatively impacted by the imposition of temperature on the breeders. So going on, the second environmental parameter in this case was salinity. Fish were maintained in land-based tanks with seawater during the 12 months from the end of the reproductive season to the start of the new one. At the beginning of which fish were evaluated, so the reproductive stage was considered. But in any case, 4 groups were formed randomly. So pairs were formed and in replicate were assigned to these 4 different groups. 1 positive control was used. So what you see as a control was also injected with GnRH at the end, while the other 3 groups were left undisturbed, but a different salinity gradually lowered was imposed to them. So the first group actually was a negative control in opposition to the first one, because the salinity was the same, but they were not treated with GnRH. So somehow we knew that they probably didn't spawn at the end. While the other 2 groups were, the fish were maintained at 20 parts of salt for the one group and 12 for the other one. During this period, fish were evaluated in terms of gonadal maturation. For female, actually you cannot see, or better, you can notice slight differences. In the control, after spawning at the end of the experiment in day 25, you can see a reduced number of heterogenic oocytes because fish were able to spawn. So you see a higher percentage of primary oocytes in the picture E. While in the negative control with salinity at 34, you can see as fish were not treated with GnRH, a higher number of atretic oocytes. As Rosa said before, a percentage of atresia is usually accepted for multiple spawners, but in this case, when a fish doesn't spawn, oocytes are usually reabsorbed. And as you can see in this picture, fish did not spawn. As I said before, it was expected, and it was also expected a higher percentage of atretic oocytes. Now, what happened in the 2 other groups where salinity was lower? It seems actually we didn't obtain at the end of the experiments any natural spawning, so salinity seems not a particular stimulus for Meagre to spawn spontaneously, at least at this moment of the study. But as you can see, the percentage of atretic oocytes, it's almost close to 0. For male, actually lower salinity, imposing lower salinity to male seems to improve spermiation capacity. In fact, the 2 groups on the right side of the graph, we found more male after the application of stripping, more male able to release sperm. So concluding, what to say about the 2 different studies? Well, for temperature, it appears quite easy to say, meaning that a constant thermal regime can affect negatively the reproductive outcome. So it's not suggested for hatcheries to impose it as a way to save money. On the other side, salinity, it's still under it's still under consideration the effect that salinity can have, for instance, on steroid production, as it's something that we are still considering. And because yes, we didn't obtain any spawning, any natural spawning, but some minor for someone, major for other effects can be seen in a positive way. So recommendation for now, it's just related to the winter thermal profile that it appears necessary for a good egg production and industrial results. This is some references I put as a important reading. And if you have any question, please. So thank you so much.
Okay, thank you very much, Stefano. As Stefano says, I don't know if you have questions, but probably you will have doubts. You can also write them in the questions and answer. Don't leave it for the end. Let's continue, and then Dinos, Constantinos, Mylonas, please. It's your turn again.
Okay. So after seeing the reproductive biology, reproductive development of Meagre, and our efforts to look at environmental methods for controlling or enhancing the reproductive process, we also wanted to look at the breeding behavior of the fish, which it's not something that has been described before, and also to look at the relation of sound production in the spawning behavior, and then identify parentage contribution of the different breeders in order to see and learn about the kinetics of spawning. We introduced sound here because Meagre belongs to a family of fish called cyanid, which are colloquially referred to as drums or croakers, because they produce sounds at different stages of their life cycle. They have a muscle in the inside part of their abdominal cavity. It's attached to the swim bladder. And by flexing this muscle, which is a muscle that has the highest frequency of contraction, it contracts with about 400 hertz. That means 400 times a second. And by contracting this muscle, they produce sound. Now, why do they produce sound? What is sound used for in this fish? We wanted to see if it's related to reproduction. We know that it's related to reproduction because we recorded fish during the reproductive season, but we wanted to know specifically how the fish they're using sound, and I will tell you at the end what was our final objectives. So my background is fish reproductive biology and endocrinology, and I'm mostly interested in describing the reproductive biology of fish in captivity and developing methods, either environmental or pharmacological methods for inducing spawning and sperm production. And I work at the Institute of Marine Biology, Biotechnology, and Aquaculture, or one of the three institutes of the Hellenic Center for Marine Research. And we're stationed here in Crete. So as you have learned from the previous two talks, the majority of Meagre reared in captivity do not undergo maturation. That is, they do not undergo the final stage of maturation in females. Males do complete maturation without a problem. And therefore, there's no ovulation, no spawning. And because of that, the majority of facilities require the use of hormones to induce the fish to mature and spawn. So in order to study the reproductive behavior of these fish, of course, we induce fish with GnRH. The typical standard protocol that has been developed involves a single GnRH injection of about 15 micrograms of GnRH in the fish, in the females, and in the males, the use of GnRH implants for the enhancement of sperm production. The specific objectives of our work was to identify stereotype behaviors that occur around the time of spawning, to monitor sound production in relation to spawning, and then to assign parentage contribution to individual breeders. Our experimental design is indicated here. We used two groups. Just a second, let me put a pointer. We used two groups of breeders. Each group consisted of 3 males and 2 females, and each group was placed in a 15 cubic meter concrete tank that is shown here. We followed the same fish for 2 consecutive reproductive seasons in order to have replicates for statistical analysis. and each tank was fitted with 2 underwater cameras, one at surveying the top of the water and the other one the bottom of the water. It's shown here. This is tank 1, top, bottom. This is tank 2, top, bottom. Each tank was fitted also with a hydrophone in order to record the sounds produced by the fish, and each fish was externally tagged. All the males got a square tag of different color, and all the females got a round tag of a different color. Therefore, we're able to identify visually each individual in the 2 stocks. As I said, we treated the fish for inducing spawning and enhancing spermiation using GnRH implants in the males and a GnRH injection in the females. So this next 3 videos show you the 3 typical breeding behavior or stereotype behavior that we observe around the time of spawning. The first we call trailing, and this involves a female, which is shown here with the round orange tag, trailed by either 1 or 2 or 3 males. These are the 2 males that are following this female at this time. This is called trailing, and as I said, it may involve 1 or multiple males in the population. The other behavior we identify is called male-to-male agonistic behavior, and as the title indicates, it's agonistic behavior between 2 males. One male will attack another male while they're swimming. And then the third behavior is called darting. And this is similar to trailing in the sense that males are following female. What you see here is all the 5 fish here. These are the 2 females, and these are the 3 males. And all of a sudden, one female may start swimming. The other one does nothing. It's not involved in this. And 1 or more of the males may be following her. This is called darting. It's similar to trailing, but it has a separate, different starting point and a more exaggerated behavior. And the final behavior we identified is, of course, spawning. Again, this is the 2 cameras, top and bottom of tank 1. And these are the 2 cameras of tank 2, top and bottom. Don't pay attention to tank number 2 because at this time nothing was happening. What you see here is one of the females being stationary in the bottom, while the other female is being trailed by the 3 males. And you will see here how they follow her very closely, and then as soon as she releases the eggs, which is this white cloud that you see, the males, all 3 males, will participate in the spawning event. However, did they all fertilize the eggs? We'll see in the following slides. So this is the ethogram of the breeding behavior, which, as I said, it included trailing of a female by 1 or more males. It included darting, again, following of 1 female by 1 or more males, starting from a stationary posture. We have male-to-male agonistic behavior, and we have spawning, which is usually involving 1 female with all 3 males in the population, so the whole population of males. Let's look at the sound now. So turn up the sound of your computer so you can hear that. We identified 4 different types of sounds, and we characterized them by the number of pulses, that they produce. Short pulses, very short pulses of up to 3 sounds, 3 pulses are called knocks. Up to 7 pulses, which are shown here, they're called short grunts. Intermediate grunts if the sound consisted of up to 29 pulses, and long grunts if the pulses were more than 29. Obviously, the brackets were decided arbitrarily according to what we observed, and this is how these sounds sound. First, the knocks. So this is the knocks. This is just 3 pulses max. This is short grunts now. you see that the quality of the sound changes. The intermediate grunts, and then long grunts. These sounds are very, very large, and in fact, you can hear them from outside the tank. In fact, you can hear them from outside the building. It's very, very large, but you have to be there at the time of spawning to hear them. And this is shown in this graph here. What we have tabulated here is the different types of sounds, knocks, short grunts, intermediate grunts, and long grunts, before induction and after induction. And then the different types of sounds produced and the number of sounds produced before induction, after induction, as a function of time of the day. You can see two important things here. First of all, that the number of sounds produced increases quite dramatically after the GnRH induction. That is, once the fish start undergoing maturation. So this tells you that these sounds are produced, or the number of sounds produced, it's related to the process of maturation and spawning. Second thing that you can notice is that although before GnRH induction, before spawning, most of the sound production was happening during the day, during the daytime, daylight hours of the day. When we induce spawning, most of the sound produced were during the nighttime, which is the time of spawning. Okay, we have also observed different types of sounds with different behaviors, but to save time, I will not refer to that now. So let's look at now at the spawning kinetics and the parentage contribution. This is the results from tank 1. The different columns are the 2 different years, 2021, 2022, and these are the fertilization success. This is tank 2. So what you see is that after GnRH injection, the fish will spawn 3 times at day 2, 3, and 4, with the majority of eggs produced at day 3. If we look now at the parentage analysis, first let's identify our fish here. We have 2 females, blue and pink, and these are the eggs produced by female blue and this by female pink. In the second year, the blue female died, therefore we substitute another female, which is female pink, and you can see here that these are the eggs produced by female pink, and these are the eggs produced by female orange, and these are produced by a female pink. You can see clearly here that in both years, the majority of eggs produced were from female pink. So there seems to be some hierarchy in this tank favoring the pink female. If you look at the male contribution, this is 100% of the eggs, 100% of the eggs, and the different colors indicate the percentage of eggs that are sired, that are fertilized by the different males. This is the percentage fertilized by male blue, this by male orange, and this by male white. Again, you can see that the majority of eggs fertilized were fertilized by the male white in this tank. And we can see that, sorry, there's a mistake here. This male was the largest male of the 3 that we put. We didn't select for it. It was just randomly this fish was selected. We selected based on the spermiation ability, but it so happened that the largest fish was the one that fertilized the majority of eggs in this tank. Let's look now in the second tank. Again, we have pink female, blue female in both years, orange, white, and blue males. This time, all males have the same size. Again, you can see that the majority of eggs produced were produced by pink, by the female with a pink tag, and the majority of eggs fertilized were fertilized by the orange male in both years in this population. This time, this male was not the largest because it was the same size as the other ones, but it was the most aggressive fish in the population. And to conclude what we found in this part of the study, of course, we have described the ethogram. We characterized the breeding behavior of Meagre during the reproductive season and after induction of spawning. So we identified the breeding behavior and the associated behaviors that take place before and after. We, as you saw, we had similar behaviors before and after, but we had a different number of behaviors, and in one behavior, which is in one sound that was produced during spawning, this was different. So we had a different sound. Females seem to exhibit a hierarchy which allowed them to spawn more eggs than the other female. Of course, we only had 2 females in the population, so it's difficult to draw solid conclusions, but we didn't want to have more fish in the tank because then it would be difficult to identify different fish, and we wanted to be able to work with the behaviors of individual fish. We also saw that males exhibit a hierarchy, which we're not sure whether it's directed by size. In one tank where different size fish existed, it was related to size. In the other tank that all 3 fish had the same size, it was the fish that was the most aggressive. So one may think that if the fish are of different size, then the big one is the alpha male, doesn't have to fight to establish his hierarchy. But in the case where the males are of the same size, then in order to establish hierarchy, there seems to be some aggressive behavior, which allows the dominant male to fertilize up to 80% of the eggs. In relation to breeding behavior in association with sound, as I said already, there's a lot more sonic activity in the time after spawning induction, so it's associated with the process of maturation and spawning. And although I did not show it in order to save time, there's also specific sounds like the long grunts that are associated only with spawning behavior. So all this knowledge is very nice, but how can we use this in aquaculture? Well, our underlying question and the hypothesis that we want to test in the future is, can we use sound playback of these sounds that we have recorded to influence reproductive function? And how do we want to influence reproductive function? For example, we can enhance maturation and we can induce maturation and therefore spawning of females by playing these sounds that are related to maturation, and therefore alleviate the need for using hormones. And secondly, can we use these sounds to disrupt the hierarchy developed by males and females, and therefore allow more individuals to participate in the spawning? Therefore, we can have a more uniform parentage contribution, and therefore the production of more families and progeny of more variable genetic background. This is our study, and we'll answer any questions when we finish with the whole seminar. Webinar.
Okay, thank you very much, Dinos. So let's continue. Let me remind again all attendees that they have the opportunity to write their questions and doubts in the questions and answer tool. Don't wait till the end. So let's move to the final presentation. This is Neil Duncan from the IRTA, Tarragona, Spain. So hello, Neil.
Hello.
Hello. Okay.
Okay, I'll share screen, yeah. Is that good?
Not yet.
Oh, yeah.
Okay, it's coming. Okay, here it is. So you can go, please.
Okay. It's a pleasure to be part of this webinar on the reproduction and broodstock management of Meagre. I'm going to talk today on the induced gametogenesis of Meagre. It doesn't seem to be changing. Okay, so I'm Neil Duncan, senior researcher in IRTA, La Rapita in Spain, working in fish reproduction for 33 years, mainly on reproductive control of species, established and new to aquaculture. I've worked in Scotland, Mexico and Spain, PhD from the University of Stirling, and working in IRTA, which is here, just in the south of Catalonia in Spain. So the objectives of this work in NewTechAqua were to introduce new biotechnologies for induction of gametogenesis, and specifically to determine the effect of recombinant gonadotropins on gametogenesis in prepubertal Meagre. The target group is Mediterranean aquaculture, where principally the area where Meagre are grown. But these technologies have wider application to other species. We've used them in other species, so it clearly has applications to world aquaculture. The summary of the content, I'll give a brief introduction, general methods of the experiment, then focus on the males, methods and results, females, methods and results, conclusion, discussion, and some kind of perspective for the future. So firstly, I'd just like to give a brief overview of the maturation process, which was excellently introduced by Rosa in the first presentation. So we have the environment signals being received by the brain, and the pituitary produces the gonadotropins, the follicle stimulating hormone FSH and the luteinizing hormone LH. These are the two gonadotropins which essentially control the entire maturation process. They act on the gonads to produce the steroids, and I'd like to point out that FSH is essentially controlling the main part of gametogenesis in the females, the vitellogenesis through to oocyte maturation, and in the males, the spermatogenesis from spermatogonia right through to spermatozoides. And the LH is controlling the maturation, the ovulation, and the spermiation. And this is the part of maturation which is commonly controlled in aquaculture with the use of hormones, as mentioned by Stefano and Dinos. The releasing gonadotropin hormones are used to induce this. So, but the initial stages with the FSH there's a less availability of hormones to control these stages, and consequently they are generally not controlled in aquaculture. And we're now introducing these technologies that can make this possible. The FSH and the LH are actually species specific, so it's important that you have to have a correct use of these hormones. You need to have the species-specific hormone, which in the case of FSH is one of the reasons there hasn't been to date an enormous amount of work and use of these hormones in aquaculture. So the idea here is to use these hormones on prepubertal Meagre to induce gametogenesis. This would give possibilities to improve reproductive control, and also can have importance for breeding programs for example, which improve growth rates by 15% generation. So if we reduce the generation time in later maturing fish like the Meagre, we can have more generations in a given period of time. So the first stage in the project was to produce the Meagre single chain recombinant gonadotropins, the RFSH and the RLH. This was completed by Rara Aves Biotech, which is a biotechnology company in Valencia, who are a part of the partnership in the project. And they sequenced the DNA sequence of these two hormones, the FSH and the LH. They placed the sequences into cell culture using Chinese hamster ovary cell culture, and this cell culture produced the hormones which they purified and concentrated and made available for the project in this experiment. The experiment was with prepubertal Meagre, 18 months, roughly 1 kilo in size. The experiment lasted 21 weeks. We had 2 treatment groups, one treatment group with the recombinant gonadotropins, the FSH and the LH, and the control group with saline injections. We initiated the experiment with weekly injections of FSH, starting with 6 micrograms, rising to 12 micrograms per kilo through 0 to 6 weeks. The LH, we used weekly injections from 3 to 10 micrograms per kilo. In the later stages of the maturation period, 6 to 12 weeks, and the control group was given saline injections each week. The work was done in a 10-meter cube tank in a RAS recirculation system. Natural photoperiod, constant temperature of 18. Gonad histology was examined on 0, 6, 12, and 21 weeks, and plasma steroids, 11 ketotestosterone, estradiol, and testosterone were measured during every 3 to 4 weeks during the experiment. So we'll start with the males. In the methods, this is the protocol of injections that we applied to the males. So we started off with the FSH, 6, 9 weekly injections, 6, 9, and then 12 through to week 9. The LH, we concentrated on the end of the maturation process with injections of LH at 9 micrograms per kilo. Blood sampling at various intervals, and fish sacrificed for gonad histology, 0, 6, and 12 weeks. So first we look at the effect of the gonadotropins on the 11 ketotestosterone. We can see in the control group from week 0 through to week 21, there was essentially no changes in the levels of ketotestosterone, whilst in the group treated with the recombinant gonadotropins, we had significant increases in the ketotestosterone. There were a few control fish that matured and had elevated levels of the steroid. If we look at the gonadosomatic index, which Rosa explained is essentially the percentage of the fish that is gonad, we can see that 100% of the males treated with recombinant gonadotropins matured with a GSI of approximately 2 at the end of the experiment, whilst in the control fish we had no increase in the GSI apart from 3 males out of the 24 that matured to have similar levels as the treated fish. We look at the testes histology. You can see in the fish treated with recombinant gonadotropins, these black dots are spermatozoides, so you can see a massive production of spermatozoides in these testes, whilst the production of spermatozoides in the control fish was relatively low. The quality of the sperm, we analyzed the quality of the sperm in 2 of the control fish that matured and all 13 treated males. And these values are similar to values you'll find in the literature for adult fish. So we had 62% motility, 2 times 10 to the 10 spermatozoides per milliliter, and good velocities. So you can see that the gonadotropins were successful in inducing maturation in these prepubertal Meagre. Moving on to the females, we used a more extended protocol of hormone application. again 6, 9, and then 12 through to week 20, and the LH towards the end of the anticipated maturation process, initially with lower doses of 3 and then 10 micrograms per kilo. Blood sampling and gonad histology. So if we look at the results from the steroids, you can see that the estradiol and testosterone, essentially there were no significant increases in the steroids. You can see that the control and fish treated with recombinant gonadotropins were essentially with no significant differences. And similarly, the gonadosomatic index, there was, as expected, no change in the control fish, but also in the treated fish, although there was a significant increase by week 21, these were all quite low values, just 0.3% of GSI indicate. And when we look at the oocyte within the ovaries, we found that the ovaries were predominantly still pre-vitellogenesis despite after all this treatment. So we saw that in these females, the recombinant gonadotropin application didn't have an effect. So in conclusion, pubertal maturation advanced 6 to 18 months in males, an increase in maturation from 12.5% in controls to 100% in the RGTH treated males. However, the recombinant gonadotropin treatment did not induce puberty in the 1 kilo 18 months old females. So we need to discuss why this is, and we suggest that the females were not close enough to puberty. The females actually mature a year later than the males in the natural maturation process, and therefore the females were approximately 2, perhaps even 3 years before maturation. So we can say that the males had developed in their testes to a stage which they could respond to the gonadotropins and mature, whilst the females the ovaries were not sufficiently developed to respond to the gonadotropins, and we need to allow the females to develop more before we can perhaps anticipate that they will respond to these treatments. Perhaps a year more, bringing them to 1 year before puberty would be sufficient to have them respond and mature at this pre-pubertal stage. And this is work that we're interested in pursuing after this project. So to look at the possible perspectives of these recombinant gonadotropins, these new biotechnologies, we've shown that you can ensure 100% maturity, and this is quite an interesting development in that you could reduce the number of breeders being maintained. If you can have confidence that 100% of your breeders will mature to produce gametes, then you could plan to maintain less breeders for the reproductive periods. We could reduce the number of years that the broodstock need to be held by 1 year, or have 1 more year when the broodstock are active, and reduce the number of years that breeders are held before they become productive for the companies. So this could amount to a saving of having to hold broodstock for a number of years. But perhaps the most interesting application could be to reduce the generation time for breeding programs. if we have a shorter generation time in a given period of time, it offers the potential to have more generations and therefore more improved growth or the trait that you're interested in over that period of time. For example, in the case of the males in this experiment, in 8 years using the RGTH, we could have 4 generations of fish. Without the recombinant gonadotropins, 3 generations would take 9 years or more. So in that period of time, we could have 1 more generation, which would imply 15% more growth for the stocks and perhaps 15% more profit. Some relevant reading. Rosa published this article on this work that I presented now. We've done similar work in flathead grey mullet, Muciocephalus, and in this work, so you can see that we produced viable gametes from both males and females and good quality eggs and larvae, and this is a good review of the recombinant gonadotropins. So, any questions, please write them through the Q&T tool. Do you want to take over, Bernardo?
Yes, thank you very much, Neil. Hello, Dinos, can you please join me for...
Yes, I'm here and available.
For coordinating the
Sure.
debate. Well, I have to congratulate all the panelists for the very interesting presentations they have done. There are very interesting results. I see that the people is a little bit afraid for writing questions. I'm sure that there are doubts, but maybe...
Let me give a two-minute summary of everything that we did, and I think it's the importance of what we did, and maybe that will give time to the audience to frame some questions. So as you saw, the problem we have with this fish in captivity is that of course, the wild fish that we bring in are not easily, they don't adjust easily to the captive environment in tanks, and therefore they need some boosting. If we want to, for example, start a breeding selection program and we want to get a variety of wild fish to introduce, to make our nucleus, then obviously we will need to enhance their maturation and to induce their spawning in order to get eggs and make them enter into the breeding selection program. Then we move to hatchery produce F1 generation fish that you saw that they have no problem in undergoing gametogenesis in captivity. The males complete maturation and they spermate quite well. By using recombinant hormones, we can advance the maturation process in males, and we can reduce the generation interval for breeding selection programs. I wouldn't recommend that, or this is not necessary for the multiplier unit of production, for general production, but it is quite useful, as Dr. Neil Duncan said, to speed up the process of domestication and selection. Then in the aspect of environmental manipulations, then our target were to use what is done in aquaculture and enhance it and improve it. And one thing that we know we use in hatcheries is constant temperature borehole water. We do this because the water is cleaner, it doesn't need filtration, and for biosecurity reasons, it's more safe. But if you use this water at constant temperature, you need to heat it and you need to cool it in order to give the annual cycle that the fish require. And this, of course, requires energy, and energy costs money, as you all know, from paying your electricity bills. So we know from another species, from sea bream, we knew that we can stimulate the whole process of reproduction and have quite successful spawning in constant temperature, as long as this constant temperature is the spawning temperature for the fish. And therefore, for sea bream, which is around 18, 19, it works fine. We know from previous studies that we can use a constant temperature during the summer in Meagre, and the fish will not only reproduce quite nicely. They don't need the high temperature of the summer, but if you keep the constant temperature during the summer, you can extend the reproductive season, and you can make the fish spawn from May until September without a problem. What we didn't know was whether by removing the winter would be something that the fish will allow us to do. And this was the target of our work, to see if removing the winter also, not only the summer, will allow us to have fish that will reproduce well. And we saw that it's not that good to remove the winter. These fish don't need the summer temperatures, but they do need the winter temperatures. Then in terms of salinity, you probably know, or for those that don't know, one of the reasons Meagre is a little bit, it's a rare fish in the Mediterranean, is because it aggregates around the mouths of the very large rivers, and they spawn in the rivers. Therefore, they spawn at low salinity. But when we rear them in aquaculture, we don't have low salinity water in aquaculture. We have full strength seawater in aquaculture. And that's why we wanted to see if by exposing the fish to low salinity during the time of spawning, we can induce them to mature. Obviously, it didn't work for females. It did seem to be better for the females in terms of not undergoing atresia so fast. And it did improve the spermiation of the males, but it didn't enhance the development of the females. But maybe we needed to do it for a longer period of time. We only did it for 3-4 weeks. Maybe we need to do it longer. So these are our objectives, and this is the results we got, and this is the direction that I want to move. And of course, with the breeding behavior and the sound, we want to try something very, very imaginable, which is to play music to the fish, but their own music, and either disturb their hierarchies and/or induce them to mature and undergo spawning. So we were hoping to learn more and be able to develop methods and technologies, both from a management point of view, but also from a pharmacological point of view, to enhance our control of reproductive processes in aquaculture for these species.
Mm-hmm. Thank you, Dinos. There are two questions in the questions and answer, but just continuing with your summary and related with these questions about controlling the reproduction process and the maturation process through the control of environmental parameters. Just let me ask you, you have worked with temperature, with salinity, but not with photoperiod in this project. What you can tell us about photoperiod manipulation or control for this species, for Meagre?
Neil, you want to answer it, or you want me to answer it?
Sorry?
I don't mind.
We didn't do any photoperiod manipulations, but I believe that it's possible to do what we have been doing has been done in all other species. That is to use photoperiod manipulations to shift the reproductive season. I'm not sure that photoperiodic manipulations can be used to induce maturation.
We did a small study, and we were certainly able to shift the period of maturation. But considering your work and some work the work of Dinos Mylonas and some work that we've done with the Canarias, if you want to have more extended production of eggs, I think the best way to go forward is to extend the spawning period with maintaining the spawning temperatures and the spawning environment, and they seem to go on for a very long extended period producing eggs, no?
Yeah.
Okay.
It's true, this will be good for maybe 5 months, but for a whole year, I would think that you will have to resort to at least two stocks, photoperiod stocks.
Yeah. Mm-hmm. It would, be advisable, yeah.
Okay. I have a question for all of you, and then it's related with the use of recombinant hormones, of the prohibition of using hormones in organic aquaculture. So, but I think that Judinos have explained it well, no? That the use of hormones has a lot of advantages. But anyway, I mean, I don't know, you can give us more light, because, well, you have well explained how hormones cannot also help to promote, ensure that the fish maturate properly and induce the spawning, but also that you can speed up the process of domestication, no? I'm sure that, I don't know, sometimes you have said that, some of you have said that it has not many sense to forbidden the use of hormones in organic aquaculture.
Yeah,
I don't know if you can discuss a little bit more about this.
Well, first of all, using hormones, we should use them only when we need them. There's no point in using them just to be fancy and just to make the process more difficult. We should use them only when necessary, because we know very well that if fish are spawning naturally, not naturally, spontaneously, it seems to be that it's always the egg quality is better. Okay, so we start from this principle that we use hormones only when we need them. And we need them in two situations. We need them in the situation where the fish do not undergo maturation, or they do not undergo gametogenesis, as we saw from Neil Duncan's work before puberty. So then we do need hormones to induce gametogenesis or to induce maturation. But also we need hormones when we want to manipulate the system. For example, if you want to do in vitro fertilization, the fish may be spawning perfectly and quite spontaneously in tanks, but they're not going to do it all at the same time, and you don't know exactly when they will do it. So you need to use hormones in order to synchronize the fish and in order to predict exactly when the gametes are going to be ready. I'm talking about eggs, so that you can strip them as soon as they're ovulated and do your selective breeding. In that case, we use it only for the breeding selection program when we need it. For the production, the general production, what we call the multiplier, you don't use it. Now, in terms of whether there's any problem with using hormones, no, we have not seen any problem with using hormones in terms of damaging the fish, unless you overstimulate. If you use a very high dose in fish that have a daily egg production, like sea bream, like puntazzo, like pagus, then you may overstimulate egg production, and then the fish will inflate and they may die. Or if you use hormones of the pituitary, which are large hormones like gonadotropins that are foreign to the fish may develop an immune response, and then maybe in following years they will destroy part of this hormone. Therefore, you will need a higher amount. These are the only problems that we have seen. So they're very rare. In terms of organic, I think there's absolutely no reason not to use them unless you're extremely adamant to the point that no, organic fish must be as natural as possible. Yeah, okay. If you want as natural as possible, you cannot use exogenous hormones. But growing them in a tank is not natural. So you immediately remove this criterion or this dogma from the point that you start feeding the fish in tanks. So in that case, then organic means nothing. And why do I say this? Because this hormone, it goes to the broodstock. It will last for 3 days. The half-life of a GnRH is only 20 minutes. It means that every 20 minutes, 50% of what has been injected into the fish has been catabolized and has been removed. So by the time the fish spawns, it probably doesn't have any hormone in its blood, and it has no hormone in the eggs. And remember, you're going to eat these eggs 2 or 3 years later. So there's absolutely no reason for me not to allow the use of hormones for organic production. But this is more of a philosophical or a political question than it is biological.
Mm-hmm. Okay, thank you very much for this clarification. I think it's very important to well, to need opinion and your position, because these things about regulation of organic aquaculture are whereas other regulations they vary it along the time when we can incorporate more knowledge to the process. So I have well, there were two questions in the questions and answers that I see that Judino has briefly answered.
The other people may have other answers though, so I would encourage both Neil and Stefano and Rosa to maybe give their opinions, especially the one that is more of a critical, which is regarding the broodstock nutrition.
Yeah.
The question in the that is very clear is that nobody's using street spawning in Meagre for production. So the fish are either in some facilities they will spawn by themselves in the tank spontaneously, or after hormonal stimulation, they will spawn quite readily in the tanks with extremely high fertilization success, almost always higher than 90%. Now, in the question about when do you need to give a broodstock diet or a careful diet for the fish, it's my opinion that at least 6 months before the spawning season. But I would welcome Neil's also extensive expertise in the area.
Mm-hmm. Yeah.
Sorry, what was that?
I was saying that in terms of the question of when is a natural, when is a good diet required for the broodstock, when do we need to be careful about broodstock diets? And I said that, in my opinion, at least 6 weeks before the spawning season.
I would prefer 6 months to a year even. It is a difficult debate because there's there's studies that show that even feeding within the spawning season can be adequate to ensure the fish have all the nutrients available that they need. But certainly I have the feeling that they need to have a reserve of nutrients, and that certainly that reserve should be there before they initiate the whole process of of vitellogenesis or spermatogenesis, which, depending on the species of fish, will be anything from 2 months to 6 months, or even a year in the northern salmonids. So I would really favor. I don't think. I mean, people do argue that giving too rich a diet can have negative effects because the fish often don't have a great deal of exercise in the conditions that they're in. But I would prefer to start earlier than later. I would be very conservative in my recommendation.
Okay, thank you. Well, I have one question for Rosa and another one for Stefano. I start with Rosa. In your presentation, in your very interesting presentation, that you did a very good review about the gametogenesis process and the maduration. At the end, you were talking about the spontaneous maturation and spawning in tanks and in cages. And according what I follow you, you have presented very interesting results for cages. What could be your recommendation to continue research on this way? Because
we need to repeat, of course, other we need to do other better study the reproduction. We need to do other sampling, because we just check for 2 consecutive years and we find a good number of males, not all the females, with post-ovulatory follicles, but we need to repeat our experiment. And then I think that we should also increase the number of sampling, because we sample fish following what is reported in the wild fish during the reproductive season, but we maybe need to reduce the time between all sampling, and so see what happens from the beginning to the end in order to be sure that, of course, the process proceeds in the best way. Mm-hmm. And then it's quite strange, or maybe more than strange, we found post-ovulatory follicles in Meagre that reproduce in sea cages which are in the sea. So with high salinity compared, of course, what is important in the wild, this is fish reproduce in low salinity water. So this is encouraging stage, but we need to straight with this to better understand if this is a reliable result. And of course, the other problem in cages is to find eggs, which of course can be transported by current, so it's not easy to check for eggs. And maybe we should think about create an experimental cage in which follow a group of fish for experimental purpose, because I forgot to say that the fish farm was just for a commercial farm. So we just took sample to fish and then analyze them. So we need maybe to plan a better a fixed experiment and to see what happens in the proper way. So this is my opinion, that we need to do more experiment and to fix the sample at the shorter way to see what happens.
Mm-hmm. Okay, thank you very much. And again, congratulations for the work done. Very, very interesting. So I had a question for Stefano, but it's related with a question already written in the questions and answer tool by Josu Cardiel. It's about the environmental variables that you have worked with. So according to your results, it seems clear that temperature is a key parameter to control and maybe manipulate when you have the fish in tanks. I didn't understand very well the results regarding the salinity. Maybe they were not conclusive. I don't know, you can say something more about that. And the question that is in the question and answer is about what other variables, apart from temperature, salinity, you think that can influence in the gonadal maturity?
Well, okay. First, starting from the result of the salinity, as you said, the study was a bit inconclusive in a way that we didn't reach, obviously, our final goal. So obtaining a spawning from untreated fish exposed to different salinity. However, as we got, in any case, some positive results in terms of capability of fish to not be subjective to atresia as when they are exposed to seawater salinity, or male having the capacity to spermiate better when exposed to lower salinity. I would say that our results need to be implemented. So probably in terms of maybe timing of exposure to salinity, either as amplitude, so the window of time that they are exposed to, or a different moment during the process of gametogenesis, before, later. That's something that has to be considered. About other environmental parameter, well, for sure, different species, they have different parameter influencing their maturation. So for instance, photoperiod. In this case, in both experiments, we decided to expose the fish for obvious reason, meaning we wanted to measure one parameter at a time. So one time was temperature, the other time was salinity. The thermoperiod that we imposed in these two studies were natural-like. So yes, artificial, but respecting the solar cycle over the year. Other parameters can be involved depending by the species. Even the pH can be important for release activation of the gametes. So yeah, in general, yeah, for Meagre, I wouldn't know what to say at the moment because we didn't test any other parameter.
Okay. Dinos, there are two questions in the questions and answer. One is from Massimo Cariano that is asking that he says that it might be possible to collect fertilized eggs from sea cages, as this has been shown to be feasible in tuna.
Okay, in principle, yeah, it is possible to do it because I assume because the eggs are quite very buoyant. So yeah, it would be possible to collect them from sea cages, I expect. And I'm saying this knowing that for amberjack, it is not so possible because greater amberjack, because we have tried that a lot and we were not very successful. Greater amberjack eggs, for some reason, are very heavy compared to other marine fish, and therefore they don't flow readily. They don't float readily to the surface, and they go out of the water column before they reach the surface, and then to be contained by the curtains. But Meagre eggs, I have the feeling, based on their floating characteristics, that they will be quite possible to collect in cages. Yeah.
Good. And there is one question. Maybe we have already discussed about this from Ahmed Saban at the bottom, asking, that it is possible to treat male squirrels in the same way as Meagre. I don't understand this. I don't know.
Maybe it was put in a translation software. It was was the name of a fish that was translated as a squirrel.
Yeah.
I'm sure Ignacio Jimenez would tell you he can develop treatments for squirrels if needed.
Squirrels. Well, if we substitute squirrel to something else, is it possible to treat male fish in the same way as Meagre in terms of reducing salinity, injecting stimulant hormones and temperature? My feeling is that if you have a fish, you can use hormones to induce spermiation, yes, by injecting the right hormones. You can use temperature and salinity maybe to enhance the process if the fish is spawning at low salinities. But whether you can induce complete maturation without the use of hormones just by using salinity, I'm not sure. I cannot tell, depending on the species that you have, but based on our knowledge or our results.
Okay. Well, one final question for you is about the fish behavior and this question of aggressiveness of the male, as this fish has been shown to be more competitive. I think that you have explained it well. It is not a question of selecting aggressive broodstock, no, but it's a question of knowing the behavior of the fish, no?
Yeah, because as you saw from the video, it's apparent that all three males are participating, and it's hard to say that one of them had a better chance, but yet in every spawn, in both years, in both tanks, one of the fish fertilized 80% of the eggs. Now, one may say, well, maybe it doesn't have to do with that. Maybe it has to do with sperm competition, because we know from other species and from sperm physiology that there is sperm competition. Not all spermatozoa are created equal. Like not all eggs are created equal. And there are spermatozoa that may be more able than others. And there are groups of spermatozoa in a semen sample. We don't know anything about that. But it seems difficult for me to believe that there's one, that the sperm of one male that is has the ability to overcome the fertilizing ability of the sperm of another male that it produces exactly the same amount of sperm, that produces good sperm, give plus or minus compared to the successful sperm, and yet his spermatozoa will be 80% more effective than the other two. So it must be something that has to do with proximity, with how close they are, in addition to maybe other aspects that we don't know. So it is very important. What we have seen, it's very important, and it's quite typical of many species, sea bass, sea bream, and this is not something that you want. You don't want it in breeding selection programs using mass spawning, because you put 20 females to reproduce with 20 males, hoping to make 50, 100 families, and at the end you have 5 families because there's only 2 males that are fertilizing the majority of the eggs. You don't want this in breeding selection programs, but you don't want it also for the production facility, because you want your fish to be of different genetic background. You want them to have a lot of variability. Therefore, they have a better chance as a group, as a population, to survive given different conditions that may be happening from year to year, different temperature profiles, different parasites, different feed ingredients, different management. So you don't want to select, you don't want clones, you don't want clones. Clones do one thing excellently, but they cannot adjust to all environments that you have outside. So you want as much variability as possible, both for the production and for the breeding nucleus.
Mm-hmm. Well, time is arriving to its end. Just one final question for you, Dinos. In your presentation, when you start, let's say, with the methodology, you say that you built tanks for the study that you done about behavior, and then you show in your videos like males and females, when they were, say, playing, they were going up to the closer to
the surface.
To the surface. Can you tell us the design of the tanks that you did?
Well, the tanks were already there. We didn't design it specifically for this, but what I must say is that if you want to do any work in this area, especially when it comes to sound, it is absolutely necessary to use concrete tanks, because fiberglass tanks, they reverberate, they're flexible, and therefore they reverberate the sound, and it's very difficult for the hydrophones to detect, just the sound produced by the fish. And also, this is number one, that you need concrete and you need to have it isolated as much as possible. And the second thing is that you have to silence your facilities. And imagine, how can you silence an aquaculture facility? And when we're doing this, it's unbelievable. You will laugh at the things that we resorted to make the facility as quiet as possible, because the hydrophones will pick up sounds from cars outside, from the road. They'll pick up the noise of the pumps from the floor, because the sound, especially low frequency sound, travels very long distances and very rapidly in dense medium, that is in concrete and in water. So these are the two suggestions that I would say in terms of whether it makes a difference to have a large tank, a deep tank, a round tank, or whatever. I tell you, Meagre is a perfect aquaculture fish. It will spawn anywhere you put it, regardless of the size of the tank. It's a very easy fish to induce spawning. It doesn't do it by itself, but once you induce it, they will spawn anywhere. We haven't seen them being really picky, as opposed to groupers and amberjack, that they need deep and large areas, but they may have to. This may have to do with their swimming behavior. Groupers, that they need to swim from the bottom to the top of the surface to release their eggs, and the amberjack, that they're pelagic, open water surface fish that require open space for their activity.
Mm-hmm. Okay, thank you very much. So I think that we have done a very good debate, and then I propose that we finish here, and then we keep the time that we were scheduling.
And remind people about the...
Yeah, and then reminded all attendees that you are invited to join us on Friday for the webinar that we are going to organize about reproduction of broodstock management of amberjack, where I'm sure that you will have the opportunity to follow very interesting results from NewTechAqua. So thank you very much to all the attendees that have been with us today, Rosa, Zupa, Neil, Duncan, Stefano, Lancerotto, and of course to Constantinos, Mylonas. Okay,
Thank you all, and we'll hope to see you on Friday.
Okay, so cheers.
Okay.
Bye-bye.
Thank you.